My name is Tatiana, and I'm Principal Scientist in vitro neuroscience here at Signature Discovery. I invite you today to join me over the coming weeks, where I will discuss you how to measure mitochondrial function. We will explore the benefits of measuring the function of this organelle, and of course, I will share with you some internal data as well. In this video, I will explore how we can use kinetics experiments to measure mitochondrial function and why this is such an important piece of unrevealing the complexity of this organelle. These assays are characterized by their reproducibility and validation and is a very important part in assessing mitochondrial function related to dynamic parameters. So actually, kinetics experiments are very important, and they can be used employing different probes that we call fluorophore. So basically, how these molecules become fluorescent when some mitochondrial function change. The convenient bit of using kinetics experiments is because we can apply all the cell types, and we can use all different plate reader formats and cell models. And we can also track mitochondrial changes over time. So here at Signature, actually we measure mitochondrial function using this kinetic experiment through different parameters. So we can measure cytosolic calcium levels, we can measure mitochondrial membrane potential, and also oxidative stress. The only difference between the assays is actually which probe, which molecule, fluorescence molecule or fluorophore, we will use in the models. On top of that, in all these kinetic experiments, we use what we call reference compound. So it's a compound that makes sure that after we use, the mitochondrial function will change. So it's a way to guarantee that the assay is working. One of these compounds is called FCCP. So FCCP is a mitochondrial protonophore, meaning that it does a lot of different and small holes in the mitochondrial membrane. And this will lead to the release of the mitochondrial calcium back into the cytosol. So let's talk about calcium then. So actually, the levels of calcium inside the cell is really important because it's associated with mitochondrial function as well with neuronal excitability and the cytotoxicity. So mitochondria is one of the most important and crucial calcium buffer systems, meaning if mitochondria is working well, it will uptake a lot of calcium from the cytosol and will accumulate this calcium inside the mitochondria itself. When we have excess of calcium inside of mitochondria, this actually can trigger cell death, and this can also lead to changes in mitochondrial membrane protection and oxidative stress. So in this figure, we can see calcium measurement using fluor3-AM as a probe. In this case, we use fibroblasts from control individuals and fibroblasts from Parkinson patients. So we can see on the right-hand side graphic that we can measure basal fluorescence fluor3. This means that we can see differences from the start between the two cell types, control and diseased. As I mentioned before, we also apply FCCP as a reference compound. And once we added this drug in the system, we can see that there is the release of calcium back to the cytosol that is represented here by an increase in the fluorescence per se. So we can visualize that Parkinson's disease cells have a decrease in the basal fluorescence of fluor3, meaning have a decrease of cytosolic calcium. So one can ask a question like, okay, why cells from Parkinson's disease actually have less calcium in the cytosol in the first place? So if we have a decrease in calcium in the cytosol, this can mean that this calcium is actually going to somewhere else. And as I just mentioned, mitochondria is one of the most important buffer systems, meaning like if the calcium is not in the cytosol, probably it's because it's inside mitochondria. So actually what we can measure as well is this delta fluor3 fluorescence, meaning it's the difference between the calcium level in the cytosol after FCCP minus before FCCP. So this can give you an idea of how much calcium the mitochondria can release back to the cytosol. And as I said before, if the calcium was not in the cytosol, it's probably because it was in the mitochondria, and this is true. So if we see this graphic on your right, actually we can see an increase in the dosolic calcium after FCCP was added. So one of the most important outcomes when we are talking about mitochondrial function is actually to measure mitochondrial membrane flotation. So as I mentioned before, the fact that mitochondria uptake this calcium, this can lead to changes in the charge between mitochondrial matrix and the membranes itself. So measuring mitochondrial membrane flotation is a very good indication of how healthy, how good these mitochondria are. So at Signature, to measure mitochondrial membrane flotation, we use another fluorophore or probe called TMRE. So using the same setup as we use to measure fluor3, that is the fluorophore to measure calcium levels, here we are using this probe called TMRE. So again, in this graphics, is represented the mitochondrial membrane flotation of fibroblasts from control individuals and Parkinson's disease patients. So at the basal level, meaning how much TMRE we do have in the cytosol, we can see that there is a decrease of the signal in the PD patients comparing to the control cell lines. One can argue like, okay, if you have less TMRE in the cytosol, where does TMRE are located then? It's very important to mention that TMRE is actually a probe that has positive charge, meaning that it tends to be inside of negative organelles. And if mitochondrial function is somehow negative, this TMRE will be allocated inside this organelle anyways. This is exactly what we see in the graphic on your right. So after FCCP, that is the reference compound used very often, these kinetic experiments, we can see that actually after it, there is an increase in the signal of TMRE in the PD cells in comparison to the control ones. Sometimes we can argue like, okay, why there is an increase of TMRE inside mitochondria if we should actually see a decrease? The fact is that, and this comes with what I mentioned in one of our previous videos, is like you have to have a whole picture of mitochondrial function to understand actually what is happening. From this graphic, someone can argue like, okay, we have good mitochondria because it's more negative, therefore accumulated more TMRE. But it's not the case because we do know that have Parkinson's cells in some models also have more number of mitochondria. But this is another story for another day. And what about oxidative stress? So how we can actually measure it and why this is so important? So in the neurodegeneration field, we know that oxidative stress is one of the reasons to actually induce cell death. Oxidative stress literally means the imbalance between of how much reactive oxygen species the cells produce and how much antioxidant the cells has available. So if you have more antioxidants, meaning that your cells will not suffer from oxidative stress. If you have less antioxidant but more ROS, this means that probably your cells will have to deal with this oxidative stress. And neurons and the brain cells in general are very susceptible to oxidative stress and ROS species inducing cell death. So actually there are a lot of functional or more physiological roles of oxidative stress, or actually physiological roles of this reactive oxygen species. It participates in cellular proliferation, neuronal pruning, oxygen sensing, and of course it's really important to respond to stress and inflammation. However, as I said, if you have an excessive production of this oxidative stress, then this can lead to dysfunction of mitochondrial membrane flotation, calcium levels, decreasing afterwards the energy production. The redox state can also damage proteins, lipids, and DNA, therefore the cells can really suffer from all this process. Here at Signature, one of the ways to measure this oxidative stress or this imbalance of how much ROS production we have is using the fluorophore called DCF or H2-DCFDA. So in this graphic, we can see the basal fluorescence of DCF, again using fibroblasts from control individuals in Parkinson's patients. So as you can see in your left-hand graphic, when we measure the basal DCF fluorescence, meaning the basal level of oxidative stress, we don't see much difference between control and Parkinson's disease cells. So from this perspective, one can argue like, okay, Parkinson's disease doesn't suffer with an increase in ROS production. And this can be true in some cases, and especially in some models. However, when we add FCCP, the reference compound, in the experiments, we can see that in this aspect, the Parkinson's disease cells is able to produce or to increase a lot of the oxidative stress that we can measure. This means that for whatever reason, the PD cell lines can actually deregulate much easier, leading to a more production of ROS than the control cell line. So again, this is why it's very important to, when we're talking about mitochondrial function, measure different parameters and have a more generic and a broad overview to understand actually how mitochondria is working. So if you missed last week's video from our series, visit our website to catch up on insights to how to measure mitochondrial function using off-the-shelf reagents and kits. And please join me over the coming weeks as I will guide you through some methods to measure mitochondrial dynamics and biogenesis. And once again, I will share with you exclusive internal data. If you have any questions, please get in touch, either in the comment section below or through our website. And visit Signature Discover website to have more information about mitochondrial assays and platforms available.